Good morning, Dr. Varghese. Welcome to my presentation. The gene of interest for me is GFP, gene acquired from Equirus victoria, which is a jellyfish, and the plasmid I selected is PUC19. Why I choose this gene and plasmid is that this plasmid is small in size, which makes it easy to handle and read, and it also contains ampicillin-resistant gene, which makes it a good choice for my cell, which is a bacteria Escherichia coli. Another reason is that this gene contains multiple cloning sites for insertion of gene. Now, why I choose GFP gene is because it is usually detectable as it emits green light when exposed to UV or blue light, and its expression is easy to detect without the need of any additional reagent. Now, to start the experiment, PUC19 can be bought by any supplier of molecular biology as well as GFP. To begin the process, we first amplify GFP gene by PCR, running it with primer that introduce restriction enzyme site for cloning. Restriction enzyme we use here is EZORI. To start the PCR process, we do the basic PCR process by mixing all the DNA template, primer, DNA polymerase, DNTPs, buffers, everything, centrifuging it, and then putting it in a PCR machine, where it will do denaturing, annealing, extension, and everything. Once it's done, as a result, we will get a PCR amplified GFP with ECORI as a restriction enzyme. After this, we do digestion and ligation. Now, what is digestion? Digestion is a process in which enzymes make a cut on the plasmid, making two sticky ends. And then after that, ligation starts. In ligation, our desired gene, in this case, GFP gene, attached to the cut area which was made during the digestion, sticking itself to the sticky ends. As a result, we see a circular DNA. In this circular DNA, we can clearly see that the backbone of this circular DNA is PUC19, and our gene of interest is attached with this backbone, which is GFP. Now it's time to put this circular DNA in our required or desired cell. In my case, it's Escherichia coli. To do this, we do the standard transformation protocol. To begin the process, we need competent E. coli on ice. Why ice? So the E. coli won't engulf their own DNAs. Once we have E. coli on the ice, we mix the mixture of DNA in this E. coli tube and leave it to rest for 15 to 30 minutes. After 30 minutes, we heat shocks the mixture at 42 degrees centigrade for 30 seconds, and after 30 seconds, we rapidly cool down this thing, this whole mixture. Once all of these steps are done, we put this mixture on the ice bag and prepare a petri dish. In this petri dish, we will choose a selective media which contains an antibiotic. In my case, it's ampicillin antibiotic. Why we choose selective media with antibiotic? It's because we only want those bacteria to grow which are ampicillin resistant. Once we have this plate, now we just incubate it after streaking the mixture on this plate, leaving it in incubator for 24 hours at 36 degrees centigrade. Once this process is done, we will see colonies grown on the petri dish. To confirm that our gene of interest has been shown in these colonies, what we do is we run the PCR again. But this time, two things will be changed. First thing is we will use GFP genes as primer, and the targeted cell will be chosen from the petri dish. After running the basic PCR procedure, denaturation, annealing, extension, when we get the result of the PCR, we run it on gel electrophoresis, and we compare our bands of the targeted cell with the original GFP genes. If it shows band, this means that it has successfully been shown in our bacteria. Another way to find out that either it has worked or not is that we expose our colonies to UV or blue light, which in this case they will emit green light, as shown in the reference image on the left. This image is exposed to the UV or blue light, and on the right side, as you can see, a typical petri dish exposed to a normal light. Thank you very much.